Hi, my name is Kyle Solberg, and today I will be talking to you about how honeybees use antenna olfactory receptors to differentiate odorants via memory association. So, a honeybee antenna typically contains around 170 odorant receptors. Honeybees utilize olfactory reception for various aspects of their life. This ranges from foraging, mating, social interactions, navigating, and more. As a result, their olfactory system is able to detect a wide range of molecules and odorants. While many of these odorants don't have any innate meaning, many can be associated with things such as food sources, danger, or landmarks. Since honeybees are generalist foragers, they must be able to detect a wide range of odorants. However, since they are typically only exposed to a finite number of plant species within a few kilometers of the hive, they must also be able to learn to distinguish between a limited number of olfactory cues. When visiting a flower, honeybees form olfactory memories of the scents of flowers that they obtain nectar from. Therefore, when they are foraging in the future and come across these same odorants, the bee is able to recall the quality of the food source and determine if it is worth foraging or not. Furthermore, because a honeybee's environment is incredibly dynamic and food sources are constantly changing, a honeybee must continuously make new memories and associations with its environment so that it can continuously and properly assess the highest quality food sources to bring back to the hive. Because Apis mellifera is the most common honeybee worldwide and is found in an incredibly wide range of environments with countless combinations of odorant cues, it is an excellent model organism to detect how olfactory receptors affect memory recall. This leads to the question, how do antenna olfactory receptors affect a honeybee's ability to recall a memory? One of the first questions scientists had was whether or not there was a difference between right antenna and left antenna olfactory receptors when forming a memory. To measure this, bees were trained using both antenna with a lemon plus sucrose solution being the positive stimulus, and a vanilla plus saturated saline solution as the negative stimulus. There was a total of 3 trials to train the bee to the positive stimulus, spaced 6 minutes apart. After 5 to 10 minutes, the bees were randomly assigned to one of 2 groups. Either right antenna, denoted RA, where the left antenna was coated with latex and rendered inactive, or left antenna, denoted LA, where the right antenna was coated in latex and rendered inactive. After 1 to 2 hours, the memory recall of the bees were tested by exposing the bees to both the lemon and vanilla solutions. This was repeated 10 times, and the proboscis extension response was scored as either A. Extension of proboscis to lemon and no extension to vanilla, B. Extension of the proboscis to both lemon and vanilla, C. Extension of the proboscis to vanilla but not lemon, or D. No extension of the proboscis. This process was then repeated 23 to 24 hours after the initial training period. It was observed that the desired A response at 1 to 2 hours was significantly higher in right antenna than in left antenna. However, at 23 to 24 hours, the A response was significantly higher in left antenna than in right antenna. This suggests that the ability of a bee to access memory shifts from antenna to antenna over time, with short-term memory recall being more associated with the right antenna and long-term recall being more associated with the left antenna. Next, scientists also investigated how well honeybees were able to distinguish between similar odors after conditioning and creating a memory association. To test this, bees were split into 3 groups, one being both antenna exposed, the other being one antenna exposed, and the final one being distal halves of antenna exposed. Each of the groups were conditioned to the odor 1-hexanol over a series of 5 trials. Each of the groups were then exposed to the training odor 1-hexanol, a similar odor being 1-octanol, and a dissimilar odor being geraniol. As expected, when exposed to the conditioned odorant, the bees exhibited the highest percentage of proboscis extension, and there was no significant difference between any of the groups. In contrast, when exposed to the dissimilar odorant, the bees exhibited the lowest percentage of proboscis extension, with the both antenna exposed group being slightly lower than the others. When exposed to the similar odorant, an intermediate percentage of bees showed proboscis extension, with the both antenna group and the distal halves exposed groups being lower than the one antenna covered group. This suggests that bees have more difficulty differentiating between similar odors than they do different odors. Furthermore, it appears that the combination of olfactory receptors from both antennae are better at differentiating between similar odorants than if olfactory receptors from only one antenna is used. Scientists also looked at how previous experience with odorants affects olfactory receptor expression in the antenna. To test this, scientists split bees into 3 groups of 30 bees and conditioned them to a stimulus. The first group was conditioned to linalool paired with sucrose in a total of 12 trials over 2 days to form a robust long-term memory of the odor. The second group was conditioned the same way, but linalool and sucrose were not paired to prevent associative learning. The third group was once again conditioned the same way, but received sucrose only to serve as a baseline. The linalool paired group showed a learning curve that plateaued at greater than 80% correct responses to linalool. The linalool unpaired group showed only a small response to linalool initially, but stopped responding by the 3rd trial. 30 hours after the first trial, the antennae from 10 bees from each group were removed, and the expression of different olfactory receptors were measured via PCR. In the paired group, only olfactory receptor 151 was significantly downregulated compared to the baseline group. This suggests that experience with an odor does regulate olfactory receptor expression, but only of the olfactory receptor binding to the respective odorant. Furthermore, the unpaired group did not show the significant downregulation of olfactory receptor 151. This suggests that associations with the odor or memory recall is necessary to regulate the expression of olfactory receptors. This experiment was then repeated using queen pheromone 9-ODA instead of linalool, and similar results to the first experiment were obtained. In this experiment, olfactory receptor 11 was significantly downregulated in the paired group, but once again not in the unpaired group, further confirming the results of the first experiment. In conclusion, there is a variation in a bee's ability for olfactory memory recall based on whether it is using the right or left antenna. The right antenna is more involved in short-term memory recall, whereas the left antenna is more involved in long-term memory recall. Furthermore, both antennae are needed for more accurate differentiation and identification of similar odorant molecules. Finally, bees that had formed a memory of an odorant with a reward exhibited less expression of the associated receptor compared to unpaired bees. This suggests less gene expression is needed for odorants with an associated memory. This research is significant because it highlights the key role antennae play in odorant identification and shows antennae are crucial for honeybees to function properly. Furthermore, it also highlights the complexity of honeybee memory, which is in direct contrast to the notion that honeybees are simple organisms. Finally, it encourages further research into honeybee memory development pathways, which has the potential of being applied to other organisms such as humans.